Dr. Selva, you can begin the introduction.
Yeah. Good morning, everyone. I'm very happy to introduce my friend and my colleague, Dr. Kapte Pradap Singh, who is currently working as a senior scientist in vegetable science at ICR, National Institute of Abiotic Stress Management, Baramati, Maharashtra. And he completed undergraduations from Mahatma MPK Rahuri in 2009 and MSc from University of Agricultural Science at Bangalore in 2011 and PhD from Tamil Nadu Agricultural University. He completed 2015. And his research specializations includes, particularly in vegetable science. There are three types of people are there, scientists. One is who are working in biotic stress, abiotic stress, and quality enhancement. And Kapte, he is only concentrating on his last 12 years. He is concentrating only on abiotic stress conditions, particularly water management, vegetable grafting, and rootstock development. So over the years, he has been actively involved for developing breeding strategies to improve the vegetable crop performance under stress conditions, particularly on the drought conditions and the waterlogging and high temperature through, particularly through grafting programs and product cultivation technologies. By his experience, and he joined ARS in 2015. He is worked in as from the several prestigious ICR institute like Kazri, Jodhpur. Then afterwards, he joined into National Institute of Abiotic Stress Management, and he has made significant scientific contributions. Not only the scientific contributions, he made very quality publications, especially in the reputed journals. Almost 50 papers, more than 50 papers he published in national and international journals, along with 9 book chapters and around 10 popular articles and 4 technical bulletins regards to by abiotic stress management. And this is research particularly on focus on vegetable crop improvement, grafting technology, deficit irrigation strategies to improve the crop productivity under challenging the environmental conditions. So we are indeed too fortunate to have such a accomplishment scientist with us today. And with these words, I invite Dr. Pradap Singh to kindly deliver your lectures. Thank you.
Um, Good morning, everyone. Thank you, Dr. Selvakumar, for introducing me. So today I'm going to deliver a lecture on vegetable grafting for mitigating abiotic stresses and enhancing resource efficiency. As your winter school is related to recent advances in protective cultivation, the scientific way towards Vikasit Krishi Sankalp. And so based on this winter school's title, vegetable grafting is a very important aspect, particularly for protective culture as well. Still, it is in nascent stage in India, but in open field, it has taken lead where a lot of area in vegetable crops is under grafting. So coming to my today's presentation, this is the content or flow of my today's talk. I will be giving a background about vegetable grafting, presenting the different aspect like world scenario as well as Indian scenario of this grafting. Then what are the principle and practices for vegetable grafting? Still, because this topic is very new, although some of the points might be overlapping with my previous speakers, because I worked in Kazri for a brief period of time before joining ICNSM Baramati. So some of the aspect like low temperature tolerance, grafting for low temperature tolerance might be repeated because Dr. Pradeep Kumar sir might have covered some of the topics. Then I will give some, if you want to go for a rootstock breeding or identifying rootstock for abiotic stresses, what sort of different genetic resources to be used, or how we can have a rootstock breeding program in brief. Then coming to like, there are a lot of scientific and technical challenges in this vegetable grafting. So I will be briefing them. And finally, I will be presenting some of the research achievements where this key grafting traits through rootstock we have studied at our institute as well as other institutes regarding each abiotic stress as a case study. Then finally, I will give a perspective of this topic in India, and I will conclude my topic. So coming to the background, as we all know that agriculture productivity must be increased by 60% to feed 9.6 billion people by 2050, since our Prime Minister have given a call for Vikasit Bharat by 2047. And in this 9.6 billion, India will be having population up to 1.7 billion by 2050, which is nearly 17 to 80% of total world population. So we need to focus on enhancing productivity, and all this productivity which we need to increase, it should be achieved through sustainable use of whatever resources we are using for producing this vegetable crops, either water, nutrients, soil, whatever resources we are using. And since we know from this century, after last 2 decades, the unfavorable or this climatic variability has increased a lot, like hailstorm, then rainfall in short period of time, drought. And if you want to take a today's example, you can see most of the parts of India, particularly in Western India, there is a heat wave, which is very which we mostly do not see, particularly in the first week of March. The climate, like temperature, has been increased, which is similar to which we see in the month of May. So it has crossed 37, 38 in most of the parts of Maharashtra and Gujarat, and so that has led to severe impact on this vegetable crops. And due to this, water resources are limited due to high evapotranspiration rate in summer season. This further challenges the sustainable production of vegetable crops. So to address all this issue, although we are using so many different technologies in vegetable, we are identifying superior varieties. We have different production technologies, either using by mulching, drip irrigation. We are nowadays, a lot of biostimulants are being used for this vegetable production. So along with all those available technology, grafting is something different and which have major share in improving the sustainability of vegetable crops. So it is a nature positive technology which is eco-friendly, and it will enhance stress tolerance. Either it is abiotic stress or biotic stress. It will improve nutrient uptake or efficiency, and finally there are resources what we are using for producing these vegetables. So coming to grafting. So what is vegetable grafting? It is a technique that involves physically joining or surgical joining of genetically different shoot and root system. So the final plant which we'll get from this grafting will be a compound single plant. And you can see from this figure, the above ground part, which is shoot, which is known as scion, and the below ground, which is rootstock, which is so grafting was mostly practiced in fruit crops. But in vegetable crops, it is coming up in a very big way. So your scion should be, so here our breeding goals are different. The scion will be bred for above ground traits, either high yield due to fruit size, number of fruits, and better fruit quality, which is consumer oriented, and tolerant to various above ground or foliar diseases. If you take, for example, tomato, leaf curl virus is a major problem, then blight is a major problem. So scion should be tolerant to all these above traits. And along with the scion, we should have a different genotype which will be used as a rootstock. It might be either a brinjal rootstock for tomato, might be tomato interspecific hybrid rootstock for tomato, or a different genotype as a rootstock for tomato. So that rootstock will possess a trait like strong and vigorous root system with superior root system architecture. It will be tolerant to water stresses, either drought or waterlogging. And since salinity is also a major challenge, which is governed by the soil quality and water quality. So the rootstock should be tolerant to this salinity and alkalinity. And in addition to this abiotic stresses, the soil-borne pathogens are a major problem in vegetable production. So the rootstock should be tolerant to these traits, either nematodes, bacterial wilt, fusarium wilt. So whatever traits which are affecting vegetable production through root system. So we cannot isolate the traits like biotic different and abiotic different, because this trait, the crops get affected at large scale in field condition. So stresses do not work in isolation in field condition. Similarly, we have to work or research this aspect using multiple stress tolerant rootstocks. So coming to the big background of this vegetable grafting. So grafting is not a new technique. It is being practiced since from centuries in fruit crops. And as far as first evidence was there, it is nearly in 1800 BC. And recently, if you take example of vegetable grafting, so it started nearly a century ago, 1920. One of the farmers in Japan used to grow watermelon in his field, and since due to monocropping, there was severe inoculum buildup in the field of Fusarium oxysporum. So what that farmer did, he took bottle gourd rootstock and grafted that watermelon on bottle gourd rootstock, and he found very good tolerance of those grafted plant, particularly for Fusarium oxysporum, which causes severe wilting in watermelon during 1920s. Since 1920, it do not have that momentum, since grafting requires sophisticated some control chambers. The rootstock should be having proper traits, rootstock traits. So it did not started in large scale during a century ago. But at present, if you see the impact of grafting, particularly in East Asia, particularly China, Japan, and Korea. So more than 95% watermelon crop area is under grafted vegetables. And similarly, the solanaceous crops like tomato, brinjal, pepper is also grafted. If you take in case of Europe, so in Europe also a lot of area is under vegetable grafting, particularly for greenhouse cultivation as well as open field condition. In greenhouse cultivation, either soilless or in soil cultivation. So in both condition under greenhouse, it is grafted vegetables are used by farmers on large scale. So if you take the adaptation of this grafting throughout the world, so nearly more than 30 countries are using grafted vegetables on large scale. And the highest adoption of these grafted vegetables is particularly in East Asia. China, Japan, and Korea are leading. And whatever cucurbitaceous crop, particularly watermelon, is leading crop in these countries. Also the Mediterranean countries like Spain, Italy, Turkey, and Cyprus, the watermelon grown in field is grafted one to address soil-borne pathogen as well as to have better nutrient use efficiency and water use efficiency. If you take the example of Japan and Korea, the cucumber which is grown in greenhouses, nearly 90% of those crop is grafted. Similarly, 60 to 70% in this tomato and brinjal, and pepper area, like particularly sweet pepper, is also increasing day by year after year. So if you take if largest concentration of this grafted vegetable are used, if you consider the European Union or Mediterranean countries, it is now used on large scale. Then it is standard practice, particularly for hydroponic system. Since the crop grown in hydroponic system, it is continuously exposed to the fertigation or like nutrient solution. So by the time it is being irrigated 1 month, 2 months after transplanting, there is a buildup of salt in case of media, soilless media, and the roots get damaged due to this high salt concentration in root zone. So the particularly if you take a case study of Netherlands, the tomato which is grown in Netherlands is 100% grafted, and the major rootstock used in Netherlands is of interspecific hybrid Solanum lycopersicum into Solanum habrocatus. So for example, I will give one example. see the vine length in protected cultivation under hydroponics, it goes up to 40 feet. So to deliver a immobile nutrient in plant system, particularly calcium, where its deficiency occurs in zinc or at fag end, like blossom end of the fruit. So it always occur at that end because it is immobile in plant system, and it is very difficult for non-grafted plants to deliver that nutrient that far, 40 feet far within plant system. So if you have a vigorous rootstock which will acquire nutrient and water more efficiently and deliver to the scion up to the 40 feet, particularly after 10 or 12 months of transplanting, the final process of the fruit will be fed very properly, and there won't be any deficiency even 10 or 12 months of crop production also. So this is standard practice in Netherlands, and the quantity of grafted seedlings is being increasing year after year throughout the world. So coming to the Indian scenario, so there was the first report of grafting from India by IHR Bengaluru, nearly after 20,201 or 2, because IHR, along with the World Vegetable Center, they have organized training of this grafted vegetable, particularly for biotic stress tolerance. Then after that, VNR seeds at Raipur, they are also pioneer in case of private sector for breeding rootstock for vegetables. And later on, after 2010 or second decade of the 21st century, there were some systemic research on waterlogging by Dr. Bhatt and their team from IHR Bengaluru. Then several other ICL institutes have started work on grafting, like NBP Research Station at Trishul, Kerala. Even there are some rootstock identified by IIVR for waterlogging tolerance in tomato. At Kadri, we have worked on identifying rootstock for low temperature tolerance in cucumber, which is a major problem in winter season for cucurbits. Similarly, TNU have worked on brinjal eggplant grafting and also other crops. In nursery also for waterlogging, it has been worked. And then CSK, Himachal Pradesh Krishi Vishwavidyalaya, there also a lot of grafting work has been done by Dr. Pradeep Kumar. So then after this SAUs and ICN Institute, this technology has been transferred or taken up by the KVKs since 2018 to 2022, like KVK Baramati in Maharashtra. Then HOE also started working, PAU, BCKV. Several like public and private institutes are engaged in grafting nowadays. But still there is and all these institutes are working to manage a regional issue. If they have issue of waterlogging, they will identify rootstock for waterlogging tolerance, either for bacterial wilt or nematode, different traits. And so they are simply using some of the varieties which are identified for different purpose, but they are tolerant to either soil-borne pathogens or this abiotic stress tolerant traits. So still they need a more systemic efforts to breed a rootstock. And among the public and private organizations, VNR Seeds have made a lot of progress in R&D of these grafted vegetables, where they have identified so many commercial rootstock, although it is in fewer crops, but they have taken them into field condition. And if you, as on today, if we consider how much grafted seedlings might be sold in India, it is more than 100 million grafted seedlings, including solanaceous crop and cucurbitaceous crops. So mostly grafting is practiced in India in tomato, brinjal, sweet pepper. These are three leading solanaceous vegetable crops. In cucurbitaceous crops, watermelon, bitter gourd, and rich gourd. So these three crops are also leading in case of cucurbitaceous crop. So mostly in fruiting vegetable crops, it is practiced on large scale. So why do we graft? So so any technology which we develop in agriculture, the ultimate aim is to have a high yield. And to get that high yield, the plant should have good vigor. And in addition to that, the whatever yield we get or whatever fruits we get, they should be of very high quality or better quality. So that is the main objective of any technology we develop in agriculture and also in vegetable crops. So this is the primary objective. And in addition to this, to achieve this objective, the stock should be tolerant to either abiotic stresses, which are mentioned here, waterlogging, drought, salinity, heat, nutrient, heavy metal. So these traits can be incorporated through rootstock in case of cyan to enhance the cyan tolerance, and also the bacterial tolerant fusarium. If you see all these traits, for example, except heat stress, most of the traits are governed by the roots. If there is a drought, the first signal will come from root. It will, due to abscisic acid, it will signal to the stomata for stomata closing to reduce the transpiration losses under stress condition. So that stress is being signaled or governed by the roots. Even salinity, the partition of salts or uptake, translocation will be governed by the roots. Heat stress, although it is above ground trait, but through grafting it can be addressed. So majority of traits which I have mentioned here, except heat, all are governed by the roots. You take bacterial wilt, the infections start from the roots. Fusarium wilt, Phytophthora root rot, particularly in sweet pepper, it is a major problem. Verticillium wilts and nematode nowadays due to monocropping, particularly in polyhouse condition, in light soil, this issue is coming up very well. And there is complex nowadays. So if you take the coastal areas, nematode and bacterial wilt is a complex. If you take some other areas, light soils of North India, fusarium wilt and nematode is a complex there. So in this way, the stresses are affecting the vegetable crops. So why we should adopt this grafted vegetable in our vegetable production system? Because it is through grafting, it is a rapid tool to enhance abiotic stress tolerance. If your cyan is susceptible, by using a tolerant rootstock, we can enhance the cyan tolerance to these stresses. It is faster alternative to breeding and biotechnical tools. See, if we are using some wild species as a donor parent in rootstock breeding, there are different challenges we face, like incompatibility barrier between cultivated species and wild species. Although, even though if it is compatible, the F1 hybrid which we get out of that interspecific hybrid, their seeds might be not viable. You can see seeds. Even the seeds will be viable, the F2 seeds won't be viable. So these issues are there, and along with the desirable traits which we try to incorporate in cultivated species background, the undesirable traits due to linkage drag also get incorporated or integrates into that hybrid. So that is one of the challenge. So if you use directly wild species as a rootstock, you can overcome that challenge, or you can use a wild species along as a male parent, donor parent, or cultivated species as a female parent. The F1 which you will get out of it, that can be used directly as a rootstock. And so it is one of the faster way to delineate the changes in the cyan through roots. And so in this way, already I mentioned about cross incompatibility barrier can be overruled if you are using directly that interspecific hybrid as a rootstock. And so by adopting this technology, most of you might be knowing a lot of fungicides, nematicides are being used by farmers in field condition to manage these stresses. And government is promoting a lot, particularly this natural farming and organic farming. So having rootstock tolerance to this trait, obviously there will be less use of chemicals. The dependency will reduce on these fertilizers, and whatever produce will be there, that will be free from these residues. And this grafting can be applicable either in field condition, although it is followed in India, but also in soilless cultivation, like European countries, it can be adopted in India also, either in soilless or soil system under protected cultivation. So these are the benefits of grafted vegetable adaptation in Indian scenario. So how this grafting will be relevant? As we know nowadays, in most of the states, the vegetable seedlings are being procured from nurseries. Even farmers are having their own nurseries. Earlier, there was soil-based nurseries. for these vegetable crops. But nowadays it has been shifted and transition has taken place in most of the states. If I take example of Maharashtra, nearly 80 to 90% farmers in Maharashtra are purchasing seedlings of these soleaceous and cucurbitaceous crops, soilless protein nursery, not in soil cultivation. So similar is in case of Karnataka, Telangana, and so many states in MP. This is coming up on a large scale. And why this seedlings we could able to produce very efficiently in protein nursery? Because most of our southern states have very good byproduct of coconut industry as a cocoa peat. So we have cocoa peat is one of the important media used for raising these seedlings. And government is promoting precision farming. Nowadays, a lot of area under vegetable is under drip fertigation and mulch combination. Drip irrigation is must. If you go for drip irrigation, the fertilization has to be applied through fertigation, and mulching is also nowadays most. Hello.
क्वालिटी इनपुट है क्योंकि क्वालिटी इनपुट तो है बट क्वालिटी इनपुट की बहुत बड़ी सब चैलेंजेस है तो सारे के सारे जो समस्या है बैकवर्ड लिंकेज की है फॉरवर्ड लिंकेज में क्या वो किसान का जो जितना इनपुट है उसको वो परचेज कर रहे हैं कि नहीं कर रहे क्या उसकी प्रोसेसिंग कर रहे हैं कि नहीं कर रहे क्या उनकी मार्केटिंग क्या उनके पास सर जो गोडाउन की व्यवस्था है या नहीं रखने की व्यवस्था सपोज एक 300 किसान का एफपीओ है और हर किसान का 1 हेक्टेयर एवरेज लैंड होल्डिंग और सभी के सभी गेहूं करते हैं एवरेज 50 क्विंटल आती है तो जो 300 हेक्टेयर से प्रोड्यूस आने वाला उसको रखने की कोई व्यवस्था है कि नहीं रखती क्या उसको प्रोसेसिंग करने की व्यवस्था है क्या वैल्यू एडिशन करने की व्यवस्था है इतना वो फॉरवर्ड लिंकेज पे स्ट्रॉन्ग एफपीओ है और स्ट्रॉन्ग नहीं है तो कहाँ पर चैलेंजेस आ रहे हैं उसको देखने की जरूरत है तीसरा सर कौन कौन से ऐसे कंस्ट्रेंट्स है सपोज फाइनेंस से रिलेटेड कंस्ट्रेंट्स है सपोज मार्केट से रिलेटेड कंस्ट्रेंट्स है सपोज इंफ्रास्ट्रक्चर से रिलेटेड कंस्ट्रेंट्स है सपोज जो हमारे एफपीओ को मेंबर है उनके अंदर आपस में ट्रस्ट ही नहीं इस टाइप की जो समस्या है उसको देखने की जरूरत है और चौथा उनका बिजनेस प्लान क्या है जब तक प्रॉपर बिजनेस प्लान उनका नहीं डेवलप हुआ है तब तक जो मेंबर फार्मर है उनका जो जैसे होने वाला है तो फायदा नहीं होगा इन्हीं 4 ऑब्जेक्टिव के साथ सर हम लोगों ने ये पूरा प्रोजेक्ट बनाया है नेक्स्ट एक दो ये सर मैं ऑलरेडी बता चुका हूँ कैसे हमने सक्सेस लिए है नेक्स्ट ये जो 4 ऑब्जेक्टिव है उसमें मेन जो हमने की कैटेगरी रखी है कि मेंबरशिप ग्रोथ क्या है सपोज हमने 20 किसानों को लेकर के एक एफपीओ बना दिया तो उसके बाद जो मेन टारगेट 300 किसान प्लेन एरिया में और हिली एरिया में 150 किसान का टारगेट को अचीव किया या नहीं किया है मेंबरशिप क्या है कितने लोग हैं जो इक्वली कंट्रीब्यूट कर रहे हैं उनकी तो ये डॉक्टर साहब आपका ऑब्जेक्टिव हो गया लोगों की सुनिए जो आपको बेनिफिट
हेलो प्रताप
Somebody is discussing it between
हेलो ये सर जो थर्ड हमारा ऑब्जेक्टिव है कि कंस्ट्रेंट्स क्या है फाइनेंस से रिलेटेड क्या कंस्ट्रेंट्स मार्केट से रिलेटेड
हेलो
गवर्नेंस से रिलेटेड कैपिटल स्टॉकवर्स का इंफ्रास्ट्रक्चर और चौथा हमारा जो उसका बिजनेस प्लान है वो किस टाइप का जो हमारे एफपीओ
Dr. Pratap, you can speak now. You are on mute. Dr. Pratap, can you unmute yourself?
Pratap, unmute and speak.
Go to Google Chrome and unmute yourself.
Keep
whether I'm audible now?
Yes.
yeah. You can continue.
Okay. Whether there is like interaction,
No. There was someone who had unmuted. I muted that person.
Okay.
Participants are requested to mute their microphone during the delivery.
So nowadays, this drip fertigation mulch combination is most popular in vegetable crops. And if you see, most of the varieties are now either semi-indeterminate or if we take our planting distance or spacing 20 years ago, it was very narrow. Nowadays, wide spacing came due to vigorous growth of these varieties, semi-indeterminate varieties. So seedling requirement has been reduced. So in this way, the area under precision farming is increasing. So grafted vegetable will be one of the important component of vegetable production in coming years. And the area under protective cultivation is increasing. So many like educated farmers are interested, along with the traditional cropping system, to enter into this venture. If you see a lot of area under like hydroponics and cultivation in increasing, and there it is benefit of longer crop duration, and then production will be very intensive in comparison to open field conditions. So in this way, and the grafting vegetables will be important component of organic and natural farming. So in this way, these grafted vegetables are relevant as of now, and their importance will increase in coming years, considering these benefits of this grafted vegetable. So coming to the graft combination, grafting can be either interspecific or intergeneric. So here I have presented some of the example of cyan and rootstock. If you take, for example, first, if you're using tomato cyan, it is Solanum lycopersicum. And if you graft this tomato on brinjal rootstock, which is having same genus but different species. So this is interspecific grafting. And the example of intergenic grafting, for example, a cucumber is grafted on pumpkin rootstock, particularly for low temperature tolerance or soil-borne pathogen tolerance. Since cucumber belongs to different genus and pumpkin belongs to different genus, although same family, but this type of grafting, which is called as a intergeneric grafting. So I have mentioned it, for example, if there is tomato cyan, which sort of different rootstock can be used? You can use a brinjal rootstock, wild brinjal, either Solanum torum rootstock, or wild tomato interspecific hybrid or F1 hybrid rootstock. So different sort of combination can be used based on the purpose, like a trait or stress you are addressing at particular location. So there are different grafting methods. In case of cucurbitaceous crops, this whole insertion grafting is most popular in East Asian country, China, Japan. And cleft grafting is also being followed. But two popular grafting methods are whole insertion grafting. It is not majorly followed in India. It is followed in East Asia, China, Japan, and Korea. We follow single cotyledon grafting in case of cucurbitaceous crops, which is also known as splice grafting. It is easy, and the chances of success is more, and more area of this tissue is being explored for graft union formation. And if you take the example of solanaceous crops, there are also so many different grafting methods are followed, but splice grafting, or which is also very popular in case of this solanaceous vegetable crops like tomato, brinjal, capsicum. So maximum area for union is being exposed in these grafting methods. That's why their popularity has been increased. So coming to the grafting efficiency, at present in India, nearly almost all nurseries are doing manual grafting, and the single skilled person can graft nearly 100 to 200. If it is having very good experience, he can graft 200 seedlings per hour. So in a day, a single person can graft up to 1,500 to 2,000 seedlings per day. But if you take the semi-automated grafting machine, nearly 600 to 700 seedlings can be grafted in an hour. So we need to have this type of semi-automated machine to enhance the grafting efficiency, because, for example, if tomato plantation season start from June to August something, so in that area, farmers' demand is very huge, very large demand will be there, and these nurseries cannot meet that demand through manual grafting. So that's why we need to have some work initiated or have semi-automated grafting machines in India. Although there have been some semi-automated machines which has been imported from Korea and Japan, but still they are not on commercial scale. So here you can see the manual grafting versus machine grafting. In right hand side, you can see some of the companies are. One of the company in Maharashtra have developed this grafting machine. So whether this video is visible, Play?
Yes, sir.
So, can we have look of this machine grafting?
ऐसे आप देते देते तो 16 स्टेशन
The sign has been cut and it has been placed on conveyor belt. They will discard that root of sign and they will keep upper portion. In next clip rootstock.
एक साइड में अपना एंड डालेंगे एक साइड वही हिसाब से भेजा और जाएगा
Now they are placing a silicon clip at grafting units. This is finally grafted to us. So now in left hand side, you can see the manual grafting. This person is placing clips on rootstock after cutting all the rootstock in a tray, and then one by one they will place cyan at graft union. So whether videos were visible?
Yes, sir, it is visible.
Yes, sir.
So coming to the grafting process, the most and important thing to have a grafted vegetable, we should have proper rootstocks. Already cyan, which are commercial varieties grown in field condition, can be used as a cyan. But for rootstock, we need to have some different special breeding programs to identify this rootstock. So once cyan and rootstock have been developed, we need to prepare seedling. This is in short how grafting is being followed. We need to prepare seedling just like our regular nursery. Then you need to make grafting. Then put into a grafting chamber for proper healing. We need to acclimatize after proper healing. We need to acclimatize those seedling in nursery conditions, and then it will be ready for transplanting for farmers. This is one of the example of cucumber grafted seedling. For example, left hand side you can see the cyan, which is 13 days after sowing, and the rootstock, which is interspecific cucurbita maxima and conchata rootstock, which is somebody is some disturbance is there. Hello. Dr. Silva? So you can see here some of the one of the cucumber grafting process has been explained. So this is left hand side is on top is cucumber seedlings 13 days after sowing, and right hand side it is pumpkin rootstock, which is 11 days after sowing. So we have sown this rootstock 2 days later after sowing the cyan to match the cyan and rootstock stem thickness. Then it has to be grafted, and this if you are not having proper grafting chambers, you can go through the small boxes for experimental purpose to maintain temperature and humidity. And finally, the grafting plants will be ready for transplantation. So here is some of the most important aspects of grafting, like graft healing and acclimatization. So these are different group of crops. For example, tomato, eggplant, pepper, and cucurbits. So on the first day of grafting, the humidity should be 90 to 100%, not 100%, but more than 90%. And that period, healing period, takes, for example, it is 4 days for solanaceous vegetable crops. and cucurbitaceous vegetable crops take some bit long time for healing, one day more. So, and during this in grafting chamber, the temperature should be between 27 to 28 degrees Celsius. It is better to maintain at 27 to 28 degrees Celsius, if not possible, but below 30 degrees Celsius. And humidity should be more than 90% for 4 days after grafting, until 4 days of grafting. Then slowly we need to reduce the humidity. It should be between 70 to 80% after 4 days, depend upon the crop cycle, which is crop species, which is mentioned here. And finally, in hardening, regular net house or polyhouse condition, you can shift those seedlings. So it will take nearly 10 to 12 or 15 days from grafting to hardening. So after that, seedlings will be ready for dispatch to the field condition. So one of the important thing here it is temperature and humidity in grafting chamber are the most crucial factors which decide the graft success rate. So these are the different types of grafting chamber. For example, in universities or institutes, we don't have large facility. We can use these plastic boxes to maintain humidity. You can spray intermittently with to maintain humidity, but the temperature has to be maintained in room condition. Then these are the commercial greenhouse, right hand side, vertically stacked, one above the other. Then below two pictures you can see these are the commercial nurseries in India following this sort of healing process using muslin cloth and this polythene cover to maintain humidity and temperature in polyhouses. So this was all about grafting process. Now coming to the rootstock breeding. So here one tentative flow chart I have prepared how we can go or develop a rootstock through this approach. So we have bred a lot of varieties since inception of this crop improvement program in different states, either at ICN institutes or State Agriculture University. Those bred varieties can be used as a germplasm. Although we have a lot of land races, particularly in case of vegetable, brinjal and other crops, those local varieties can be used as one of the source or genetic material, and wild species can be also one part of this. So this is the different genetic base or germplasm resources to be used for rootstock breeding. So either you can have directly inbred line from crossing, or this will act as a core collection after shortlisting trait-specific germplasm from different group. So this germplasm we need to screen for the desirable traits, either a single trait. For example, I have mentioned here water stress or drought. You can screen either for individual trait or multiple traits. If you want to have a rootstock tolerant for multiple stress, you need to first screen all the germplasm for individual stresses like drought, waterlogging, then bacterial wilt, fusarium wilt individually. So after screening the germplasm individually, you need to see the common genotypes having tolerance to multiple stresses. So those genotypes having tolerance to multiple stresses, those can be used for breeding a rootstock for multiple stress tolerant rootstock. Or if you want to go for individual stress, you can use a rootstock which is tolerant to individual stresses. So after identifying those lines, we need to enhance these selected lines. We need to identify, we need to make inbred lines out of that, either crossing them with some wild species, or we can use directly one of the inbred line as a cultivated species and wild species as a donor. Directly F1 hybrid rootstock can be developed out of that crossing. So after developing inbred line tolerant to particular trait or as a rootstock or F1 hybrid rootstock, you need to test those developed rootstock for its compatibility with cyan, because we found that most of the time not all rootstock are compatible with all the cyans, because we have a lot of varieties in case of tomato, brinjal, capsicum, cucumber. So it is not necessarily each and every rootstock will have better support the cyan irrespective of genetic background. So after testing that crop compatibility, rootstock cyan compatibility, the best winning combination has to be taken into further. You can test that graft combination either for different traits like yield, morphophysiological traits, its biochemical profiling under stress condition, in greenhouse or field condition. So, because we will be having n number of rootstock tested with n number of cyan, to narrow down them, you test them first in greenhouse condition. After identifying some shortlisted graft combination, you take those to the field condition along with your non-grafted as a control. So after screening in field condition, the rootstock performing better for trait of your interest, that will be one of the new product will be developed or achieved through this breeding program. So this is in short flow chart. It can be modified as per your convenience and based on your experience. So some of the donor species which can be used for rootstock breeding, particularly in case of tomato, Solanum pimpinifolium, which is ancestor of cultivated tomato, Solanum habrocatus, then Solanum pennelli is there, Solanum chilense are there. So these species have a lot of abiotic or biotic stress tolerant genes, which can be used as a donor parent to develop interspecific hybrid rootstock, F1 hybrid, or you can identify some lines in F2 population. Then these are the some of the wild species of eggplant which we have tested as a rootstock for eggplant grafting under water deficit condition. For example, Solanum gilo is there, Indicum, Macrocarpum, Cicentrifolium torvum. So coming to the some of the scientific and technical challenges which most of the nursery growers are facing in India, because this is mostly nursery-based seedling production. Farmers cannot directly go through this opportunity, but some farmers group can come and have their own grafted nursery or like this way. For example, there is a very high cost of rootstock as of now in India, because only one or two companies are there, or they have identified systematically rootstock for tomato and brinjal. So since there is no competition in the market, for example, if you take a brinjal rootstock which is being used for tomato, for nematodes, bacterial tolerance, the individual seed cost is 1 rupee to 1 rupee 20 paisa. So if you take this cyan seed of tomato F1 hybrid seed, which is non-grafted, it is hardly 40 paisa, 40 to 50 paisa. So you can see the difference between the cyan seed and rootstock seeds. So that will add the cost of the grafted seedling. So we should have more players in the market which are breeding rootstock for different traits. So when there will be competition, the seed cost of rootstock seed cost will get reduced, and the grafted seedlings will be more affordable to the farmers. Second challenge is that if you are using brinjal rootstock or wild species as a rootstock, the sowing time of cyan and rootstock will be different. For example, Solanum torvum, it is very slow growing wild species. So it needs nearly more than 2 months to achieve grafting stem diameter for tomato. So it has to be sown 2 months before sowing of cyan. And if you take a brinjal rootstock, so to have brinjal rootstock for tomato cyan, it has to be sown 30 to 40 days before, 4 to 5 days before sowing of tomato seeds. So in this way, we need to adjust the sowing time. Similarly, for capsicum and hot pepper, because hot pepper rootstock is being used for sweet capsicum. So then the third point is that already I mentioned previously that all rootstocks are not suitable for all size. After identifying different rootstocks, we have a lot of different varieties in the market, or our public and private sector. So somebody, can you mute yourself? So this we need to identify the winning graft combination. For example, I mentioned that all rootstock are not suitable for all sand. So after testing them, their graft affinity, success percent, their performance in green and field condition. After that, only the rootstock has to be released in the market. So grafting is techno-intensive skill. Not all laborers or manpower can do grafting efficiently. So if there is unskilled manpower, the success rate will reduce. These are some technical challenges. We need to have a proper control environment to maintain that temperature and humidity. If those conditions are not maintained, in improper healing condition, the mortality will be very high. And even skillful person should have very proper knowledge of aligning the cyan and rootstock in manual grafting. So these are some of the technical challenges we have found that the nursery growers face while producing the grafted seedlings. So until now, I have covered about the background of this grafting, challenges, and different aspects of importance of this grafting in Indian scenario. Now coming to the second part of this presentation, I will be showing some of the cases which for different traits. As of now, working on individual stresses, rootstock tolerant to individual stress. But recently we have initiated multiple stress tolerance rootstock improvement by combining abiotic and biotic stresses. So here we can induce or improve the cyan tolerance to all this trait through rootstock. For first and foremost is high vigor and yield, cold tolerance, particularly in North Indian plains. The cucurbitous crops are warm season crops. Their growth will be severely affected under low temperature condition. Then compact canopy, particularly for mechanization. Then flood tolerance, then pathogen resistance, drought tolerance, salinity, heavy metal stress, and heat tolerance. So the traits which I have highlighted in green color font. So those traits I will be presenting as a case study, and we have worked on this trait either at Kazri or at our institute at NIASM Baramati. So coming to the first important trait, after screening different wild species of eggplant, brinjal, we identified 2 rootstocks having very good water use efficiency under. As we know, in semi-arid and arid regions, water stress is one of the major challenges in India. So here, in first stage, what we did, we have screened this rootstock under field condition at by withholding the irrigation water for 2 months. Similarly, we have tested them in greenhouse condition also by shortlisting genotypes from greenhouse and field condition. Only wild species as a rootstock. Without grafting, we have screened them. So we have shortlisted 5 species out of 13 wild species we have screened. And after shortlisting those 5 species, at second stage, we have tested their graft compatibility and performance in greenhouse condition in pot study by withholding irrigation water for 20 days after as compared to control. So then after identifying their performance based on morphophysiological and biochemical traits, we were interested to find their performance under field condition for yield and water productivity. In third stage, we have taken this field trials from 2001 to 23 for 3 years in field condition under deficit irrigation. So we have screened different rootstock. This below left-hand side picture, you can see the brinjal or eggplant grafted on different wild species rootstock. And this is how in pot study we found that under withholding irrigation for more than 20 days, the wild species try to allocate more biomass to the root as compared to shoot. So root to shoot ratio get increased under stress condition if you're using wild species as a rootstock. And finally, we have tested them in field condition, and we found that one of the accession of Solanum torrum and Cisimbrifolium, they have very good performance in field condition and the yield increment was nearly 20 to 41%. So that the yield increment by using Solanum torum rootstock was 22%, and by using Sesquipedalium rootstock it was 41%. So, but a challenge was that the Solanum Sesquipedalium rootstock is highly spiny. So, so that was the major challenge for grafting. Even though it is having very good yield increment up to 40% over non-grafted under 40% water deficit condition, but Solanum torum is somewhat less spiny and more amenable to the graft for brinjal. So that rootstock can be promoted over the Sesquipedalium rootstock. So these are the biochemical traits, physiological traits, particularly with respect to canopy cooling and photosystem 2 efficiency. So in left-hand side figure, we have grafted Solanum melangena on Solanum gilo, Solanum indicum, macrocrapum sesquipedalium, and torum rootstock. The last one was the Solanum melangena non-grafted, and we have studied the canopy temperature under control and stress condition. So we found that under stress condition, the Sesquipedalium and torum rootstock were able to maintain cooler canopy as compared to some of our first 3 rootstock, even as compared to non-grafted. And when we studied this photosystem 2 efficiency, which is one of the most important indicator of abiotic stress, through chlorophyll fluorescence imaging, leaf chlorophyll fluorescence imaging, we found that the Solanum Sesquipedalium, Solanum melangena and Sesquipedalium graft combination and Solanum melangena and torum graft combination were able to maintain significantly higher quantum yield. It was more than 0.7, whereas in case of 10 days or 20 days after stress withholding irrigation, the efficiency has been significantly reduced in non-grafted, even some of macrocrapum and indicum rootstock. But after this stress period, we have irrigated these grafted plants to see the recovery performance. So recovery was also better in case of Sesquipedalium torum, also in case of gilo. But some of the rootstock and non-grafted plants were not able to recover properly after that 20 days of drought period. So in this way, these grafted plants were able to, due to their more allocation of root biomass, they were able to efficiently acquire the moisture available in the soil profile and able to maintain proper water balance with cyan by having proper plant and soil interaction, water balance. So in field condition, what we found that we have imposed 3 different irrigation level. One is as a control 100% of crop evapotranspiration, then 80% of crop evapotranspiration. So water requirement was there. We have supplied 80% of that crop water requirement, where 20% deficit was imposed. And the severe deficit condition, 60% of crop evapotranspiration. Means in last you can see we have applied nearly 40% less water, whatever under normal growing condition crop requires. And different graft combination and the non-grafted plants were tested for water productivity, which is kg per meter cube. And we found that as the level of deficit increases, the water productivity was increased because less amount of water was used by the plant to produce yield. So among severe deficit condition, we found that Solanum Sesquipedalium and torum were significantly having significantly better water productivity as compared to non-grafted and even some of the other wild species. So this is the importance of using this wild species as a rootstock. You can see the performance of this morphological or phenotypic performance of these grafted plants under 20 days of withholding irrigation. You can here 3 different blocks are there, plates, and in each plate left hand side is a control plant where no stress were imposed, and right hand plant is 20 days withheld with irrigation, which is water deficit. And we found that this grafted plants on this rootstock were able to have more root to shoot ratio as compared to non-grafted plants, and also their respective control plants. So in this way, this Sesquipedalium and torum rootstock have performed better even under greenhouse conditions as well as field conditions to enhance this drought tolerance or water deficit tolerance and enhance the yield and productivity of eggplant. So if you want to know the more details, this paper I have already published in Applied Water Science. You can go through this paper. So you can see the under stress condition how the root to seed ratio has been increased. So that was about the brinjal case study. Coming to another study we perform on tomato to enhance the water productivity. Since we have shifted to soilless cultivation over our open field condition or soil cultivation in the polyhouse condition. So we have now shifted more to, or we are exploring the more water efficient technologies. So Dutch bucket production system is coming up in big way, but we need to have optimized this production practices for Dutch bucket production. And can we find some benefit of using grafting or grafted seedlings in Dutch bucket production system that we have studied in this cherry tomato experiment. So we have used 3 different scion. Pusa cherry tomato 1, which is red color variety. Pusa golden cherry tomato 2, it is yellow color cherry tomato. And fully J3 is one of the red cherry tomato variety bred by MPK Ravi. First 2 were bred by IRI CPCT, New Delhi. So, and we have used 5 different rootstocks, wild species as a rootstock, tomato wild species as rootstock, along with control. So, in that SLR 9, 57 are Solanum bipinifolium, and SLR 52, 54 are Solanum habrocatus, and SLR 66 is Solanum peruanum. So this study was performed under Dutch bucket production system, and we have recorded different morphophysiological fruit biochemical traits, root parameters, and finally the water productivity. In this way, grafting has been done for this tomato seedlings, and this was the performance of crop in Dutch bucket production system. So I will directly present the final most important aspect of study, like yield. So when we have used 3 different scion along with different rootstock and non-grafted, we found overall the SLR9, one of the Solanum pimpinellifolium rootstock, performs significantly better over the non-grafted overall, irrespective of scion, and which is nearly 40% higher over the non-grafted plants. And if you compare the performance of individual variety on different rootstock, we found that Pusa golden cherry tomato, we have coded it as a PCY. It was having significantly very high yield compared to red color varieties, and that yield was achieved better on this Solanum SLR9 rootstock, which is Solanum bipinifolium. So we can find that it's nearly up to 2-fold higher yield in grafted plants over the non-grafted plants. So we found most of the yield increment was there in case of this Pusa golden cherry tomato variety over the red color variety. And also, if you consider the rootstock performance for red color variety, it was better for pimpinellifolium based rootstock over the habrocatus or the perennium rootstock. So yield increment was better in this rootstock due to their root system architecture. So under hydroponics condition, continuous, already I mentioned about the salinity buildup is one of the major challenges in this soilless cultivation. And the water temperature increases a lot in the soilless media, because this Dutch bucket is circulating, completely closed and circulating system. So here you can find the green color bars, orange color and purple color. So green color is of Pusa cherry tomato red 1, Pusa golden cherry tomato, this is coded as PCY, and Fully J3 is red color variety. So you can find the graft combination with SLR 57 or SLR9 rootstock having significantly higher root dry mass per plant over their respective non-grafted plants or other graft combination in each set of scion. So here you can see the first row in different scion is of SLR9 rootstock, then SLR 52, 54, 57, and this one is Solanum perennium, and the last one in each row is their respective control. So when we studied the improvement in water productivity, so we found that the performance of rootstock in improving water productivity is not similar for all rootstock. So overall, the improvement was significantly higher in Pusa golden cherry tomato, then followed by Fulle Jayasri and Pusa cherry tomato red. And in that also the pipistrellum based rootstock as well as one of the Abiotic rootstock have performed better over the non-grafted plants. In this way, the grafted plants on wild species rootstock, although these rootstocks were not interspecific hybrid, but we have initiated work on making some crosses using this best performing rootstock as a scion, has a donor parent to have F1 hybrid rootstock, which will be which will be testing their performance of parents and F1 hybrid rootstock for Pusa golden cherry tomato in coming season. So the previous two case studies on eggplant grafting and cherry tomato Dutch production system was exclusively for enhancing the water productivity. And some of the open field studies in cucurbits I'm presenting here from Italy on watermelon grafting. So in this you can see two pictures on the top. The left hand side picture is of watermelon grafted and right hand side is non-grafted one. And under three different deficit irrigation level, 50, 75% and 100% of crop evapotranspiration in grafted and non-grafted. So in grid one of the watermelon variety is grafted on PS 1313 rootstock. So there was very good yield increment or biomass production per plant in grafted plants. But when we see the yield increment in grafting, it is nearly 60% more yield was achieved in this grafted plant or non-grafted plant, irrespective of stress condition. And the water productivity was also significantly higher over the control condition and in deficit irrigation. So what made these grafted plants to yield 60% better over the non-grafted plants? So higher water uptake ability of roots due to the rootstock were able to explore the root zone or soil profile very efficiently due to its different genetic architecture of architecture, and it has resulted into better CO2 assimilation in the soil due to maintaining proper balance, water balance in the rootstock and soil under deficit irrigation. Also improved the nutrient uptake. When they studied the iron partitioning, metal partitioning, mineral partitioning in cyan and rootstock, they found the grafted plants have superior or more nitrogen content, potassium and magnesium content in cyan over the non-grafted plants. So due to under stress condition, the better signaling of ABA from roots have efficiently controlled the stomatal regulation and reduced the water loss in grafted plants, where the case not there in case of non-grafted plants, and that has resulted into better intrinsic water efficiency. Also, the agronomic as intrinsic water efficiency was higher in case of grafted plants. So the about three case studies were about of water deficit conditions in solanaceous and cucurbits crops. This case study I will be presenting on flooding stress. As we know, particularly some of these states, Madhya Pradesh, Maharashtra, Gujarat, and even some other parts of Telangana, due to sudden heavy rainfall in short period of time. You can see I have displayed one picture over here. So there is a one event we found that 70 mm of rainfall in just 3 hours, and this field of tomato has nearly 40% losses after since water was stagnated for more than a week. So 50% crop was damaged. So what makes these non-grafted plants lead to 50% damage under flooding condition? Since due to waterlogging, there is reduced oxygen diffusion rate in case of soil that create hypoxia. Under flooding condition, the root respiration decreases, that creates anoxia in plants, and due to this heavy downpour, there is either nutrient leaching will be there or nutrient uptake or translocation will be affected under flooding stress. So to address this stress, we have studied tomato grafting on different rootstock, and here we tested some of the brinjal rootstock as a tomato wild species, which are mentioned in my Dutch bucket cherry tomato experiment. So we were interested to know that whether already there was some of previous reports from IHR using Solanum melongena as a rootstock for improving waterlogging tolerance. But in these conditions, in semi-arid region, the soils are highly saline, having EC of 2 to 3 decicement per meter, and also water is also saline. So what happens when there is a severe downpour in case of Kharif season, the soil salinity is more and the waterlogging will be created. So that is not a normal waterlogging. So waterlogging in saline condition. So that study we found here, so if we impose waterlogging with high saline water, what will be the performance of this ion? Whereas in previous studies, there was water done for waterlogging in case of tomato, which were for normal water condition. So this was the grafting process we followed, and this was the methodology in pot condition what we followed. Like we have grafted tomato on Solanum melongena and tomato wild species. We are allowed them to establish for 30 days after transplanting, and we have maintained one set of the treatment as a control set, and the another set as a waterlogging. So after 30 days, we have exposed this plant for 10 days of waterlogging to see the which rootstock were able to survive even 10 days of saline waterlogging conditions. And we have seen the recovery of these grafted plants after 40 days. So during this process, we have studied different physiobiochemical traits, starting like SPAT value, which is an indicator of chlorophyll content, then proline, and the MDA, RWC. So we found that the under flooding condition compared to control, these parameters, particularly chlorophyll content, chlorophyll was reduced, and there was significant enhancement of proline under stress condition as one of the tolerance mechanism of plant under abiotic stresses. So there was cell membrane damage, lipid peroxidation was there. So most of the treatments under waterlogging of different graft combination as severe membrane damage was observed. And you can find in last bar, which is non-grafted, which was significantly higher over the this tomato rootstocks as well as brinjal rootstock. So I have coded this as a Solanum lycopersicum, Solanum melongena 1 to 7 rootstock, then Solanum lycopersicum into Solanum pipidifolium 1, 2, 3, then Habrocatus 1, 2, and Penelli, and last one is non-grafted. So under stress condition, we found that the brinjal rootstock in first set from first 1 to 7 were able to perform significantly better over the eggplant, wild tomato wild species rootstock and non-grafted plants. So under stress condition, this relative water content in case of brinjal rootstock was significantly better over the tomato wild species rootstock as well as non-grafted. So in this way, the plants, grafted plants on brinjal rootstock were able to maintain proper water balance, because in waterlogging there will be similar effect will be exerted as like of drought. Water uptake will be affected, root respiration will get decreased. Under those conditions, how plant will able to acquire, translocate the nutrients and water to sign under those stress conditions, and that's what efficiently better in Solanum melongena rootstock. So we have studied the photosystem 2 efficiency, and it was better significantly. There was no difference in photosystem 2 efficiency in control and non-grafted plants, even 10 days of waterlogging. You can see the first set in brinjal, where some of the tomato wild species rootstock under waterlogging condition, the efficiency was reduced significantly, and also it was significantly lower in case of waterlogging condition in non-grafted plants. So this is the comparative performance of the plants in different graft combinations. The top row is Solanum melongena grafted rootstock grafted tomato grafted on melanjana brinjal rootstock, and below it is tomato wild species rootstock, and this last one is control. And the left hand side pot is of control condition without any waterlogging stress, and the right hand side pot is of 10 days of waterlogging. The EC of that water was nearly 2.5 decimn per meter. So you can see when waterlogging and salinity in combined exert a lot of effect on under combined stress condition. And among those, we found only 2 rootstocks, Solanum lycophersin grafted on Solanum melangena 2 and 3 genotypes. Even 10 days of the saline condition, they were able to have 32 survival, 30% survival. So the grafted plants on these 2 rootstock have twofold higher yield compared to non-grafted plants, and the survival rate was up to 30%. Whereas in case of other rootstock of brinjal and tomato wild species, there was 100% mortality. So what made these grafted plants in these 2 particular rootstock to have better survival due to better chlorophyll content, maintaining photosystem 2 efficiency and water strength status, and it has low oxidative damage due to membrane lipid peroxidation and better photosystem 2 efficiency even under stress condition. And when we studied the root biomass and all these traits in this rootstock, the root biomass was better in this 2 brinjal rootstock or the other tested rootstock. That has enhanced their flooding resilience along with the saline water for sustaining the tomato production in semi-arid regions. So that was the about flooding. And now third case study I will be presenting on salinity. It is a major problem nowadays in case of semi-arid and arid regions. So 20% irrigated area is also affected. So why salinity is a major problem in this arid and semi-arid region? For example, if I give an example of Baramati, our long-term average rainfall is 560 millimeter. But if you take into consideration how much water is lost through evapotranspiration, it is nearly 1800 mm water is being lost through evapotranspiration. So there is nearly 1000 mm deficit is there from precipitation to evaporation. So that creates the salinity. The salts available in below ground, it comes on the top layer under stress condition. So response of salinity will be in 3 different ways, either osmotic, nutritional, or toxic. But one of the benefits we found of this drought and salinity stress, the total sugars and quality traits enhances better under these 2 traits. Although there is some bit advantage of this stress, but yield reduction is severe. So how crop performance is affected the salinity condition? So due to osmotic effect. So even water is there in through irrigation we are applying, but deficit arises due to low water potential of soil solution and the soil induced nutritional disorder. So since in salinity condition there is a lot of sodium and chlorides in the soil or irrigation water, so their uptake will be more as compared to uptake of potassium, and there will be competitive uptake of the sodium and potassium under stress condition, and the uptake, transport and partition of the sodium, chloride and potassium will vary based on the genotype or irrigation method. So it will disturb the cellular homeostasis due to this toxic ions, and that will damage the cell membrane when the concentration is more in the plant. So how rootstock can act as a biological filters to address this salinity issue? So what happens if you have rootstock bred for salinity tolerance and your regular cyan is being used through grafting? So increase root or root to shoot ratio, the more salts will be. So rootstock will have one ability. They will not. They will have more beneficial ion uptake rather than harmful ion. So they will try to exclude sodium or chloride at root level due to their transport channels. So each species have different affinity or translocation or transporters for this ions, sodium or chlorides. So that first check will be there at root level. exclusion will be there. So second point, so whatever salts the root will acquire, it will not allow it to translocate to the shoot. So those salts, toxic ion will be restricted to shoot, and there won't be any damage to this ion or cell membranes or lead marginal burning, which we observed under salinity stress. So that effect we found of cyan. And so even some salts are being translocated through graft union. So due to vigorous canopy of cyan, those salts will get accumulated in case of vacuoles of this cells. And if more canopy is there, their compartmentization or particling will be better as compared to non-grafted plants. And also some of the rootstock have better potassium uptake over the sodium. So that will create less toxicity to this ion and leads to better under salinity condition. So one of the case study I will be presenting on eggplant grafting. So the rootstock which we have used for deficit irrigation to enhance water property, same set of rootstock we have tested for salinity condition in pot, where we consider 0.5 as a normal irrigation water. We have enhanced irrigation water salinity up to 3 deciml and 6 deciml. And when among the different wild species we have tested, we found Solanum macrocarpum as one of the rootstock having better nutrient or ion partitioning and better efficiency. You can see in case of control, as the level of salinity increases, the growth reduces. Similar was the case in case of grafted plants. But one thing you can see from these pictures, that the toxicity was more in case of non-grafted plants at 6 deciml compared to non-grafted plants in 6 deciml. So toxic metals were more absorbed, sodium and chloride, and less potassium uptake was there. That has led to toxicity, and you can see the yellowing and leaf fall was there in case of higher salinity level, whereas that effect was not there in case of grafted plants on Solanum macrocarpum rootstock due to mechanism which I explained in my previous slide. And what we found that the chloride, what made these grafted plants on macrocarpum rootstock to have better growth and efficiency and less toxicity. So they have excluded nearly 30% less chloride was there in the root as compared to non-grafted plants, and shoot 60% less chloride was there under 6 deciml per meter. And the potassium uptake was better. Here the potassium uptake was less, and in root we found more potassium uptake, like nearly 40% higher in root and 50% higher in case of shoot. And the potassium to sodium ratio was also better. That has nullified the sodium uptake, which is one of the toxic ion. And here it was less potassium to sodium ratio. So this is the result of whatever I explained in my previous slide. As the salinity level increases, the sodium content also increases, chloride uptake also increases. The potassium uptake reduces under salinity condition, and the balance between potassium to sodium also reduces as the salinity level increases. But one thing you can see here that the Solanum melangella grafted on Solanum macrocarpum rootstock has very less chloride uptake in case of shoot over the non-grafted plants, and even in other rootstocks under severe salinity condition. That has led to better potassium uptake over the other combinations and non-grafted plants, and it has maintained higher potassium to sodium ratio. So that was the reason which the grafted plants under severe salinity able to maintain proper growth and physiology and elevated the salinity stress. So coming to the another study of suboptimal temperature on cucumber, this study was done at Kazri, Jodhpur. Since in winter season, particularly December, January, the temperature goes below 10 degrees Celsius, and being a warm season crop, the growth is significantly affected in case of cucumber. So we have tested different cucurbitaceae species like bottle gourd, bitter gourd, sponge gourd, and different species. Even pumpkin we have tested, and we found one of the rootstock, particularly Cucurbita fusifolia, one of the landrace of fig leaf gourd, cultivated at Ooty region in Tamil Nadu in low temperature condition, has a better rootstock for grafting this cucumber on this Fissifolia rootstock. So we found that when we graft this cucumber on this Cucurbita Fissifolia rootstock, since what happens under low temperature conditions, the physiological processes slows down in plant, and ultimately the growth is very slow under low temperature condition in case of warm season crops like cucumber. So we found that after grafting, the fruiting was early by nearly more than 2 weeks in grafted plants. The yield increment at the end of cropping cycle was up to 40%. This has led to better leaf area and also photosynthesis, higher shoot and root biomass. That has led to better fruit set and fruit number, and also the fruit weight. Individual fruit weight was better over the non-grafted plants. For more details, you can explore this paper by Kumar et al. In 2019. And when we studied the nutrient content, the potassium, calcium, particularly this macro nutrients were better. Also the iron content in this grafted plant. That has led to better yield in this grafted plant under suboptimal temperature condition. Then this case study is about the nutrient use efficiency in muskmelon. This study, we have very less work on studying using rootstock to enhance the nutrient use efficiency. So this study we have initiated in our programs list program, because as of now, whatever rootstock we have identified for drought or waterlogging, same rootstock we are testing them for this nutrient use efficiency. Because under stress condition, those rootstock were able to explore the proper soil profile and nutrient uptake. So there might be some of the benefit for enhancing nutrient use efficiency under stress condition. So this case study by Kola and co-worker in 2010, although this study is very old, but very good study was done in Italy. And nowadays we can find that there is a 3F crisis: food, fuel, and fertilizer. So food is a major challenge. Since due to geopolitical situation, having fuel is also major challenge, and the fertilizer transport is being affected under this crisis. So we need to have a rootstock having better resource use efficiency. So Protean is one of the muskmelon variety which is ungrafted. You can see in the right top panel, and lower panel you can see the same variety was grafted on this pumpkin rootstock, and different nitrogen was applied starting from 0.5 millimole to 10 millimole, up to 15 millimole. So we found that as the level of nutrient increases, the growth increases. But which what made this grafted plants to enhance the growth under even under nutrient deficit condition? If you consider the 2.5 millimole of nitrate nitrogen applied in non-grafted plants, similar dose applied in grafted plants, you can see the vine length is very higher, which is up to 2-fold higher compared to non-grafted plants. So what made improvement in nitrogen, particularly nitrate nitrogen use efficiency? You can see in field condition also they were tested by applying different nitrogen doses up to 120 kg per hectare. Even non-applied control was also tested in grafted and non-grafted condition. So even when we do not apply even a single granule of fertilizer to the soil, there was 18% increment in case of grafted plants in marketable yield. And so it is not necessary if you go on increasing the nutrient fertilizer, the yield will increase. So the increment was reduced as the level increases. So to maintain the proper soil nutrient balance, if you see the 60 kg per hectare nitrogen, the 40% yield increment is one of the better when we do not apply any fertilizer. And when we go for higher doses, the yield increment is less. So these doses are better. We are applying optimum fertilizer and also getting better yield in case of grafted muskmelon. So what made these grafted plants to enhance the nutrient use efficiency? Due to better root density. So more numbers of roots were there in grafted plants, were able to explore nutrient from this rhizosphere and that has led to enhanced cyan vigor, which I have presented in my previous slide. But in case of field, found that the endogenous hormones were particularly auxins, and more intensive assimilation was there to amino acids and protein that led to better yield in grafted plants over the non-grafted under different nutrient doses. So these case studies were all about the different crops, but recently some studies are also going on, basic studies of transgrafting. So if we do not have any of the source to breed rootstock, since a lot of um, many countries are not able to accept transgenic crops, particularly in case of edible crops like vegetables. So genome editing has come up in large scale, where we are not adding any foreign genes of any of the organism. So similarly, if you can breed or develop a rootstock which is genetically engineered, genome edited, rootstock can be used under different condition, but still the study is in lab scale. But there are good hopes, particularly for biotechnology expertise, to develop a genome edited rootstock. Then coming to rootstock, so whatever we have presented, different case studies of different stresses, abiotic stresses. So these were all governed by rootstock mediated. So overall picture is that what made this cyan or this vegetable crops enhance this stress tolerance through grafting. So more vigorous root system of the rootstock has led to better nutrient water uptake, better plant water balance, shoot and root plant water balance, signaling under stress condition. That has led to improved water and nutrient uptake. So due to that better balance, water balance, the photosynthesis efficiency of grafted plants was better due to proper water relations. It has also better, stronger antioxidant defense mechanism. Under stress condition, whatever ROS were generated, reactoxin species were generated, that were scavenged very efficiently through this rootstock mediated approach. Better upregulation of hormonal signaling, having better intrinsic water efficiency. And there are also some reports which has revealed that in addition to all this morphophysiological biochemical signaling, there are some the molecular signals like large and long distance movements of mRNAs or sRNAs and proteins through graft union of rootstock to cyan. Led to improvement in the cyan performance under stress condition. So this also need to be exploring when we develop rootstock for multiple stress tolerance. So whatever case studies I have presented, so we have taken this rootstock to the field trials. You can see the in first trial, because we need to rootstock, we can't directly give them to farmers, but we have convinced some of the nursery growers, if you try this grafted brinjal over the non-grafted, it has better harvesting window, better tolerance to soil-borne pathogen, water productivity, fertilizer use efficiency. And we have found that under summer season, the grafted plants on Torum rootstock have less infestation of these red mites. So it is a major challenges in high temperature and low humidity condition. So in the middle picture, we have tested this rootstock for waterlogging tolerance along with bacterial trait, these two traits in field conditions. And in last slide, you can picture you can see that farmer standing, which is in front of him is non-grafted plants which has wilted. Behind him, the next row back to him is of grafted plants, which is green when survive after waterlogging and wilting conditions. Recently, one of the case research I didn't presented in this topic, because it was exclusively for abiotic stresses. But whatever rootstock we are identified for waterlogging or abiotic stresses, we are testing those rootstock for bacterial wilt also, because in Kharif season, along with waterlogging, this wilting due to soil-borne pathogens, either nematodes or bacterial wilt, is a major challenge. So one rootstock we have identified for waterlogging as well as bacterial wilt. We have tested in this recent last Kharif season, and you can see very clear difference of these grafted plants and non-grafted plants. In middle pictures, the left-hand side is a rootstock which is tolerant to waterlogging. Brinjal rootstock used for tomato, which is waterlogging tolerance and also tolerant to bacterial wilt. So since bacterial wilt strains are very virulent based on the different states, particularly in Kerala, the bacterial wilt strain are very virulent. Even in Maharashtra, it is different. In Chhattisgarh, it is different. In Odisha, it is different. So we need to test the rootstock for multiple location, and it is not necessary that rootstock which is tolerant to bacterial wilt strain available in Maharashtra, it will be tolerant in Chhattisgarh. So for that, we need to have multi-location trials. So you can find the right-hand side row is completely wilted, which is non-grafted. Left-hand side is tolerant. The eggplant rootstock tolerant to bacterial wilt and flooding on left-hand side, whereas non-grafted plants were completely susceptible and succumb to bacterial wilt under rainy season. So coming to the perspective of this today's talk, so we need to develop more affordable healing chamber, because since very high-tech structure like fan pad polyhouses are required to maintain temperature and humidity, but we have found that if grafting is done in small structures, even in particularly in month of July, August, when temperature will be somewhat between 30 to 32 degrees Celsius, and humidity levels are naturally will be high in rainy season. So in those sorts of healing chambers also, the farmers can go for grafting at their field also. So we need to have indigenous grafting machine and automation to reduce the labor cost and improve uniformity, graft uniformity, because the demand of grafted seedling will be more. It will not be made through nursery. We need to go for atomization, scaling of this grafted plants production. Although very regional specific here and there rootstock breeding is going on in India, but we need integrated large scale national program by involving multiple stakeholders, state agriculture university in different states, also ICER institutes, also private sector, to work in integrated way to develop rootstock in vegetables. We need more understanding, particularly as far as molecular signaling is concerned, how rootstock mediated mechanisms improves the science test tolerance and vigor. Then physiological and we need more understanding about also genetic and physiological basis of this graft mediated tolerance, either abiotic stress tolerance. And since during the process of grafting, the grafting seedling cost is more. For example, tomato grafted seedlings cost nearly from 8 rupees to 12 rupees, depends upon the location, nursery, state, and normal grafted non-grafted tomato seedling cost up to 1 rupee 50 paisa or 2 rupees. So there is nearly a very high cost of grafted seedling. So we need to optimize. Can we use less number of seedlings? Because the vigor of scion and the architecture will be very different of grafted plants. We need to standardize the agronomic practices, because the agronomic practices which are being recommended or developed, which is for non-grafted vegetables. So along with this grafting research, we need to optimize the agronomic practices of these grafted vegetables also. Then how this rootstock influence the fruit quality and quality attributes. So we found that when we graft tomato and brinjal rootstock over the non-grafted plants, the shelf life of grafted tomato is very better compared to non-grafted tomato. Also the mineral content, particularly potassium and magnesium content, was more in grafted tomato on brinjal rootstock over the non-grafted. So there is a response from farmer that taste of grafted seedlings grafted tomato is much more better. And we found that the tomato which is being transported from Maharashtra to Uttar Pradesh, the grafted tomato have better shelf life compared to non-grafted plant. It has been absorbed by the vendors itself in Uttar Pradesh when the tomato was transported from Nasik, Maharashtra to Kanpur in Uttar Pradesh. Then enhancing the grafting use efficiency. So how we can convince the nursery growers, the field management, and farmers can the benefit of grafting. Then only we can show some demonstration field trials after realizing the field yield increment or yield reduction in yield losses. Then only this farmers or nursery growers can enhance or scale up this grafted vegetable production. Then already I mentioned about biotechnology approaches like transgrafting and genome edited rootstock. It can be one of the new perspective. Then coming to the conclusion of today's class or lecture on this winter school, as you know, vegetable grafting is a sustainable technique for enhancing productivity because we will be using less resources to produce more, or we can stabilize the yield. It can address the grand challenges posed by the abiotic or biotic stresses. Abiotic stresses due to climate change, seasonal rainfall, heat stress, drought. Biotic stresses, since pathogens are evolving, if you have bred some variety, we need to change that variety. That variety won't be in market in vegetable, particularly in case of vegetable, for 4 to 5 years or 6 years, or hardly 7 years. Why? Because that variety becomes susceptible to the either nematode or bacterial wilt or fusarium. If you take, for example, bacterial wilt, the pathogen is evolving in field. If you bred variety tolerant to bacterial wilt, the pathogen will find different way to survive, and that resistance will break down. New pathogen resistance will evolve, but genetic constitution of variety will be same. So we need to also have a different genetic constitution, or having different support of rootstock and sign, different genetic background to improve that tolerance. Obviously, it has improved. We have seen the case study where the resource use efficiency is significantly enhanced, either water or fertilizer being used. It will reduce the dependency on use of chemicals. Pesticide, already I mentioned pesticide means in case of brinjal, the mites problem is less. Otherwise, a lot of miticides are being used to manage this mites in summer season. Also, fertilizers will be less, chemical use will be less, because farmers are using a lot of fungicides to reduce the damping off, or fumigant are being used to kill this bacteria under wilt conditions. So in this way, the chemical usage or dependency will be less. The water produce will get, it will be residue free or having less residues compared to non-grafted plants. And since enhancing yield is a very challenging thing, even we can stabilize the yield and reduce the yield losses, that will be the great achievement at present. So that will sustain the yield if you're using some this better rootstock under this climate change scenario. So this is all about my today's lecture on vegetable grafting to mitigate abiotic stresses and enhance the resource use efficiency. So here is thank you to everyone, particularly organizers, for inviting me and for this lecture on vegetable grafting for this winter school participants. So thank you. Silva, I have completed